AFLP, it is also called as AFLP PCR or simply AFLP, because this technique it can be used in combination with PCR. जैसा कि हमने RFLP को डिस्कस किया, तो इसमें इनिशियली जो था वो PCR का यूज़ नहीं था. सिंपली हम DNA को किसी सोर्स से एक्सट्रैक्ट करते थे और उसकी फ्रेगमेंटेशन करते थे बाय यूजिंग सर्टन टाइप ऑफ एंडोन्यूक्लियेजेस, और उसके बाइंडिंग पैटर्न को हम रिजॉल्व करते थे. लेकिन सम टाइम क्या होता है कि जो हमारे पास क्राइम सीन से जो सैंपल आता है, वो उसका जो टिश्यू है या ब्लड है, उसकी कंसंट्रेशन बड़ी कम होती है, तो उसको ज़्यादा टारगेट करने के लिए, एम्पलीफाई करने के लिए, वी हैव टू यूज़ द PCR technique. So in AFLP we are using the PCR so that we can enhance the sensitivity of the procedure. And this technique was developed in early 1990s, and it also finds its application in the field of genetic engineering or genetics and molecular biology. Now here, what is the main difference when we compare it with RFLP, or restriction fragment length polymorphism? So it is also used in the same manner to study the variation within the DNA sequence, that is polymorphism in the DNA. But here, like the RFLP, the DNA, it is fragmented or digested by using restriction endonucleases, and after that the digested fragment that have sticky ends, they are joined with adapter molecules, and adapter, they are having the known sequence. Now if you remember that previously we study the difference between adapters and the linker molecules, कि like adapter है तो उसके sticky end होंगे, एक end blunt होगा, and in case of linkers, both ends they are blunt, but they have the ability to ligate it within the DNA fragments. Like adapters, they are also having one end sticky, so it can bind with the sticky end DNA fragments after its digestion. अब इसका एडवांटेज ये है कि अडेप्टर का सीक्वेंस नोन है, तो हम क्या करते हैं कि नोन प्राइमर सेट यूज करते हैं फॉर इट्स एम्प्लीफिकेशन. सो because of this reason that we are going to amplify the restriction fragment, it is called as amplified restriction length polymorphism. And after amplification, now we are going to separate the different fragments on the gel. Now this technique, it is having more value as compared to first two techniques, because in first two techniques the sequence of the DNA was not known. So that's why in case of rapid we are going for a random amplification, and in case of RFLP we are not going to use the PCR initially, rather the bending pattern, it is resolved on the gel after restriction. But here in AFLP we are increasing the sensitivity of the reaction by using the PCR. So this technique, it can also be used to find the variations within the DNA sequence or the polymorphic DNA. So like other two techniques, the same application is there, that it can also be used to find the relationships between different organisms, like genome mapping can be performed, and the relationship between different organisms can be elucidated, and disease linkage can be elucidated by using this technique. So when we compare it, rapid or RFLP, the sensitivity, it is relatively high.